Good morning all of you. Today I delivered my seminar in my college. My research topic is biodegradation of industrially important textile dyes by actinomycetes. My research guide name is Dr. Anil Narsinghe sir. Now let's talk about my topic. My topic introduction is the in our country there are too many textile industries, so they are produces more dyes in our effluent. They are used for coloring cloth, very dangerous dyes. Azo dyes they are used. Azo dyes are used in various industries. These dyes are useless after use. Let the different water bodies such as river, lakes, these dyes are not easily degradable the environment and causes carcinogenic, mutagenic, toxic to aquatic life. The environmental pollution caused by release of industrial wastewater with the wide range of azo dyes serious problem in the present days. Azo compounds contribute the largest and the most diverse group of synthetic dyes that are widely used. A number of industries such as textile, food, cosmetic and paper printing, azo dyes containing one or more azo bond minus N is equal to N minus account for 60 to 70% of all textile dyestuff used. These dyes are recalcitrant to the biodegradation due to addition to toxicity. Let's talk about the actinomycetes. Actinomycetes is a gram-positive bacteria. They are commonly found in the soil. Colonies of the actinomycetes from the fungus-like branches, not easy to culture and isolate. Actinomycetes usually 1 to 2 diameter. My project or my research topic objectives are collection of sample from textile industries. I collected samples from Ichalkaranji in Veerbhadra process, Raghunandan process, 2 or 3 textile companies. I also collected dyes. My objective, in second objective, is the engagement of dye degradation in actinomycetes, isolation of the dye degradation actinomycetes, identification of dye degradation actinomycetes. My working plan is the collection of literature from the internet and other sources. My first plan of working is the collection of literature, articles from the internet and other sources. Collection of dyes. I am also collected dyes from Veerbhadra process and Raghunandan process. That name was yellow C4G, navy blue BNC, yellow M4G, red M5D, reactive or orange M2R from various industries. Collection of water sample from the Amit process also, Veerbhadra process also, Vishnu process also, Anand process also, Raghunandan process also. That industries is located at the Ichalkaranji, India, and I am collected sample on 2nd January 2023. These are some pics. I am collected dyes from these textile industries. There is the effluent. They are waste. That company wastewater is this. They were very harmful for the human bodies. They are directly through the drainage directly into the drainage. Next. In last 6 months, I am also poster presentation in Goa, Satara, a part of as an international conference. I am also attending the international conference on environmental sustainability and biotechnology 2022 opportunities and challenges at Goa. I am also attend and poster presentation RUSA sponsored international conference on emerging trends in applied microbiology, food science 2022 at Satara. These speakers are the poster presentation in Goa. I am presenting their poster presentation. these are some also pics. Other my research topic methodology is the study will be conducted. I'm Department of Microbiology, KSK College Build. I'm using for my isolation culture media is mineral salt medium, what we call starch casing agar or actinomycetes isolation agar. Sample I was collected in contaminated water sample and effluent and contaminated soil were collected from the Amit process, Biodegradation process, Vishnu process, Anand process, Rabandhan process, textile industry, Currently, samples were used as a source of isolate bacteria with distinct morphological colony characteristic capable of decolorizing selected azotides. The contaminated water and effluent sample were collected in airtight bottle and filtered through ordinary filter paper to remove the large suspended particle, and the filter we are used for the isolation of actinomycetes. Yellow C4G, navy blue BNC, yellow C4 M4G, red M5B, reactive orange M2R dye was collected from the textile industry currently India. All chemicals are used in highest purity and the analytical grade. Next slide. The actinomycete will be also isolated from sample collected from waste disposal site of textile Amit process, Biodegradation process, Vishnu process, Anand process, Rabandhan process, Currently India by enrichment culture technique. I am using enrichment culture technique for isolate the samples from these textile industry. The sample collected will be subject to the enrichment culture technique. The enrichment will be carried out in 100 ml mineral salt medium containing 0.1% yellow C4G dye taken in 250 ml enriched flask by adding the 10 ml of each sample separately. The flask will be then incubated in the rotary shaker at the 100 rpm at 30 degrees for 1 week. Isolation and characterization of biodegradation actinomycetes. A loopful culture from the enrichment flask was treated on the sterile mineral salt medium agar plate actinomycetes isolation agar with 0.1% yellow C4G was incubated at the room temperature for 1 week. Similarly, effluent sample will directly straight on sterile M5 MSM agar plate with 0.1% of yellow C4G dye and incubated room temperature for 1 week. The identification of the dye degrading actinomycetes strain will be carried out based on morphological culture and physiological culture characteristic of colonies obtained on plates. Observation and results I have found MSM agar plate containing 0.1% yellow C4G dye will be observed for the growth of actinomycetes and its dye degrading potential. The isolate organisms which have the ability to degrade dye in the identified based on morphological culture and physiological characteristic. I am also attending one of the science exhibition and 2023 at KSK College Build. There are some pictures and their supervisors are there present. I am isolating the actinomycetes from the soil. These are some Google Maps pictures. Next slide. I would like to thank Dr. Anil Singh for his continuous and guidance and support for my research work. He gave me the inspiration to do the project in biodegradation. He really helped me to understand the importance of the collection of sample property in the isolate way. I would also thank you to Dr. Gulwe Madam and my research center KSK College Build. These are my references. Okay, thank you.